Good morning student. Today discuss about that BioBotany chapter 9 tissue and tissue systems. So tissue and tissue systems. The chapter introduced the internal structure of higher plants. So that introduce the higher plant internal structures. So first morphology, you see that all the morphology character in outer character structures. But these chapters we discuss about that all the internal structures. Internal structure means stem, even not only for parts in tissues levels also we can see that these chapters. The study of internal structure and organizations of plant is called plant anatomy. It's called plant anatomy. Plant have cells as the basic units. So plants have a cell. Cell is a basic unit because cell group of cell is called tissues, group of tissues is called organ, group of organs is called system, group of systems is called plant or body. So basic the cell is a basic unit. The cells are organized into tissues. The tissues in turn organized into organ. The different organ in the plant have different internal structures. It is studied the dissections of microscopic examinations. The tissues. What is tissues? The tissues is a group of cells. The tissues is a group of cells that are like in origin, structure and functions. The study of tissues is called histology. So they will ask two more questions. Histology means the study of tissues. It's called histology. A plant is made up of different type of tissues, or there are two principal groups. One is meristematic tissues and is permanent tissues. So meristematic tissue is nothing but it is divided. So it is growth on unlimited growth is divided in that cells. But permanent tissues they cannot divide. So it's a permanent. That is called permanent tissues. Meristematic tissues. Now we can see of the meristematic tissues. Characteristics and classifications. The characters of meristematic tissues. Merist means divisible. It's called divisions. So this is called meristematic tissues. The terms meristems was coined by C. Negley in 1858. They will ask one more questions. The meristematic cells are isodiametric and they are maybe oval, spherical or polygonal in shape. They are meristematic tissues. They are different shapes are there. Oval, spherical or polygonal in shape. They generally have dense cytoplasm with a prominent nucleus. So the cell is generally they are cytoplasms and a prominent nucleus are present in the tissues cells. Generally the vacuoles are either small or absent. So vacuoles also is there or absent or small in size. Their cell wall is thin, elastic and made up of cellulose. These are most actively dividing cells. It is these cells are most actively dividing. So it divisions. They are cell divisions. It will do it. Functions are there in these tissues. Meristematic cells are self-perpetuating. So what is classifications of meristems? It is a meristematic tissues. So there are three types of meristems. You can see that have been classified into several type of basis of positions, origin and functions of divisions. First one, theories of meristems, organizations and functions. Many anatomists illustrated the root and shoot apical meristems on the basic the number of arrangement and accordingly proposed following theories. So that root or stems whatever. So it will arrange and accordingly proposed and following theories. An extract which is discussed below. First one, shoot apical meristems. So they are apical cell theory. First one is apical cell theory. Shoot apical meristems of apical cell theory. Apical cell theory was proposed by Hofmeister in 1850. This questions also is very important. They will ask 3 more questions. That all the questions are very important chapters. These lessons supported Hofmeisters 1852 and supported by Negley in 1859. A single apical cell is the structural and functional unit. So it is called apical tissues, apical cells. So single unit is the functions of theory. These apical cells governs the growth and the development of whole plant body. The apical tissues are developed into whole plant bodies are developed. It is the applicable in algae, bryophytes and in some pteridophytes also, that all the plants, algae, bryophytes, pteridophytes, that all the plants apical cells growth on the development of whole plant body. That is called apical cell theory. Next one, histogen theory. Histogen theory is proposed by Hanstein 1868 and supported by Strasburger. They will ask one more questions also. So very important that theory also. The shoot apexes comprises of three distinct zones. One is dermatogens, peribulum, second is third one is plerome. That is called, you see, that is called dermatogen regions. That outer layer, that middle is a peribulum, and third inner layer is called inner regions is called plerome. That is called histogens. Dermatogen, it is the outermost layer. It gives rise to epidermis. It's developed into the epidermis. Peribulum is a middle layer that gives rise to cortex regions. The plant cortex regions are produced this peribulum in that regions only. Plerome, it is the innermost layer which gives rise to stele. Stele means it is nothing, it's nothing but it's a vascular bundles. Xylem and phloem also is called stele. Pith also is called stele. So three regions, that is called histogen theory. Is very important for that scientist name also. That name also they will ask 3 more questions. That all the theories are asked under root and shoot theory also asked to 3 more questions. Very important. Next, tunica corpus theory. Tunica corpus theory is proposed by A. Schmidt 1924. The two zones or tissues are found in the apical meristem term. That is, one is the tunica, another one is corpus. So that is called, outer layer is called tunica, that innermost regions is called corpus. Tunica. The tunica is the peripheral zone. So peripheral means outer layer. So peripheral zone of the shoot apex that forms epidermis, that forms in a epidermis. The corpus, it is the inner zones of shoot apex, the form cortex and the cells of shoot. So what is difference between histogen theory and tunica corpus theory? So histogen theory is a 3 regions. One is dermatogens, peribulums and pleromes. That but tunica corpus theory only for 2 regions. One is tunica and another one is corpus. That is 2 regions only there. That is very important. That also shoot theory, to shoot apical meristem theory. That next one, root apical meristems. Root apex is present opposite to the shoot apex. So root apex is present opposite to the shoot. Shoot opposite, the present opposite theory is called root apex. The root contains root cap is the apex and the apical meristem is present below the root cap. It's called, that is called root cap. So below the root, in below the root cap, it's called root cap. The different theories proposed to explain the root apical meristem organisms are given below. One is apical cell theory. That also apical cell theory is proposed by Negley. The single apical cell or apical initial composes the root meristems. The apical initial is tetrahedral in shape. That is called tetrahedral in shape and produce root cap from one side. So one side is a root cap and the tetrahedral shape. The remaining 3 sides produce epidermis, cortex and vascular bundles. That is called vascular bundles. The 3 regions found in the vascular cryptocams. Next one is histogen theory. Histogen theory is proposed by Hanstein 1868 and supported by Strasburger. The histogen theory is applied to the root apical meristem. Speak for 4 histogen in the meristem. 4 histogen. They are respectively dermatogen, peripulum, plerome, calyptrogens. The another one is calyptrogens. The extra dermatogen is the outermost layer. It is gives the root epidermis. Dermatogen. See, that is called termogens, epidermis. That is called termogens, dermatogens. Outermost layer, it gives the root epidermis. Peripellum, the middle layer, it gives rise to cortex. It is called. That is yellow color is the no. That is called cortex regions. That is called peripellum. Third one, pleurum. The innermost layer, it gives rise to steel. It is called. That is called steel region. That is called three regions. Another one is calyptrogens. The four regions are the root. Calyptrogens gives rise to root cap. Next one, Corpor capi theory. Corpor capi theory is proposed by Scoop. There are two zones is root apex, corpor and cap. There are two regions are there. Corpor zones forms the body. Cap zone is form the cap. This theory is equivalent to tunica carpus theory. So equivalent to tunica carpus theory of shoot apex. The two regions divided and distinguished by the type of T. T is also called Y divisions. So shape of Y divisions. Corpor is characterized by inverted T divisions. Capi is straight T divisions. So corpor is a inverted T divisions. Capi inverted T divisions. Capi regions. There are two regions are there. That is corpor character is inverted T. The divisions. Capi by straight T divisions. Next ones. Quiescent center concept. Quiescent center concept was proposed by Claus in 1961 to explain root apical meristems activity. The center is located between two cap and differentiating cells of the root. The apparently inactive regions of cells in root meristems is called quiescent centers. It's called quiescent centers. That region is called the center. It is called quiescent center. Inactive regions. It is the site of hormone synthesis and also ultimate source of meristematic cells of the meristem. That is called root apex of cell theories. So very important that one, root apex cell theory and apical cell theory. It is very important. The diagram also very important because sometimes ask diagram only. That is classifications of meristem depend on the positions, origin, functions, plan of division. They are classified into 4 types. That apical meristem position is here 3 types positions. That is called, one is upper region is called apical meristems. Middle region is called intercalary meristem. That down regions is called lateral meristem. Depend on the positions. There are 3 types. Apical meristem, intercalary meristem, lateral meristem. Apical meristem present in the apex of root and shoot. So both are same regions only. It is responsible for increase the length of the plant. So length of the plant or increase, no, that is very important. Responsible for these regions. These regions are divisible, so divided and develop into height. They will not develop divided, they will not develop into height. So that is very important. So that height, the plants, some plants are very height, no. That reason that apical meristems is all divided in fast. So that only will give so height. So that it is called primary growth. Intercalary meristem occurs between the permanent tissues. It is responsible for elongations of internode. Internodal elongation. Internodal elongations means that one region, nodal region, 2 nodal regions, the in between gap, that is called elongator. That is called intercalary meristem. Lateral meristem occurs along the longitudinal axis of stem and root. Responsible for secondary tissues, thickening of stem and root. So thickening the size of increases. That is called stems and root. Example, vascular cambium and core cambiums. Next one, origin. Primary meristem, secondary meristems are there. Primary meristems derived from embryonic initials and differentiated into primary permanent tissues. It's called permanent tissues. Secondary meristem, it is derived during later stage of development of the plant body. Produce core cambium and interfascicular cambiums. Third one is functions. Depend on the functions based on the three types: prototerms, procambium, ground meristems. Prototerms, it gives rise to epidermal tissue systems and develop into epidermis, stomata and hairs. They are all developed into these regions. Procambium, it gives rise to primary vascular tissues. Example, xylem and phloems are developed into these regions. Ground meristem, it gives rise to all tissues except epidermis and vascular strands. They will not develop into it. So all the parts are into ground meristem. Plan of division: mass meristems, rib meristems, plate meristems. Mass meristem is divided in all plants. Example, endosperms, young embryo and sporangiums. Rib meristems divided anticlinally in one plants. Development of cortex and pith. Plate meristems divided anticlinally in two plants. So development of epidermis. That is positions of classifications of meristems. Permanent tissues. The permanent tissues develop from apical meristems. They loss of power of cell division. Here is no day cell divisions. So only for permanent tissues, either permanently or temporarily. They are classified into two types. One is simple permanent tissues, another one is complex permanent tissues. Simple permanent tissues. Simple tissues are composed of one type of cells only, that is called simple permanent tissues. These cells are structurally and functionally similar. They are structurally and functionally are similar. They are three types: parenchyma, collenchyma and sclerenchyma. First one is parenchyma. Para is beside any hints to pore. Parenchyma is generally present in all organs of the plant. So all the organs are plants in all organs are present in the parenchyma. It forms the ground tissues in the plant. Parenchyma is a living tissues and made up of thin-walled cells. So it is called parenchyma tissues. So thin-walled tissues, cells. The cells walls made up of cellulose. Parenchyma cells may be oval, polyhedral, cylindrical, irregular, elongated or armed. They are different type of cells are there. The tissues normally has prominent intercellular space and may store various type of materials like water, air gastric substance, occasionally store resins. Occasionally parenchyma cells which store resins, tannins, crystals of calcium carbonate, calcium oxalate are called idioplast. You see that parenchyma is a intercellular space. They have more prominent intercellular space are there. So in the intercellular space, the different kind of materials like water, air substance are there. So occasionally parenchyma cells store resins, tannins, crystals of calcium also present in the intercalary substance. That is called idioplast. They will ask two more questions. Idioplast. Parenchyma is the different type of, some of them are discussed below. So different types of are there. Arenchyma, storage parenchyma, stellate parenchyma, collenchyma and prosenchyma. The diagram also is very important. So different types of cells in parenchyma. They will ask 5 more question types of parenchyma cells. First one, arenchyma. Parenchyma which contains air in intracellular space. Air. The intracellular space is occupied the air. It help of aerations and buoyancy. So example, Nimbia hydrilla. That Nimbia hydrilla plants are buoyancy methods. They are water in up surface. They are floating because the reason the arenchyma tissues are present in the Nimbia. You know that that's why that Nimbia and hydrilla plants are floating in the water. Next one, storage parenchyma. Parenchyma stores food materials. Example, root and stem tubers. That potato also tubers, no. That also parenchyma tissues. That is called storage parenchyma. Next is stellate parenchyma. Star-shaped parenchyma. Example, petioles of banana and canna. That is called stellate parenchyma. Next one, chlorenchyma. Parenchyma cells with chloroplast. Parenchyma cells with chloroplast are present. That is called chlorenchyma. The functions in photosynthesis. Example, mesophyll of leaves. All the leaves are contain chlorophylls, that is called chlorenchyma. Next one, prosenchyma. Parenchyma cells become elongated, pointed and slightly thick walled. It provides mechanical support, that is called prosenchyma. So parenchyma tissues are 5 types of parenchyma. Next one is collenchyma. The collenchyma is a simple living mechanical tissues. Collenchyma generally occurs in hypodermis of dicot stems. It is absent in the root and also the petioles of pedicels. They are absent in root, petioles and pedicels. They are absent in collenchyma. The cells are elongated and appear polygonal in cross sections. The cell wall is unevenly thickened. They are not evenly. So the cells are unevenly thickened or there. That is called uneven. The cell wall also are not same like that. So different types of cells are there. Contains more of hemicellulose and pectin besides cellulose. It provides mechanical support and elasticity to growing parts of the plant. Collenchyma consists of narrow cells. It has only few cells, small chloroplast or none. Some of the collenchyma, some of the chloroplasts are there. Tannin may be present in the collenchyma. Tannin, that based on the pattern pectinations of the cell wall, there are 3 types of collenchyma. Type 1, angular collenchyma, lacunar collenchyma, laminar collenchyma. The diagram is also very important. They will ask 5 more questions. Collenchyma, types of collenchyma. Angular collenchyma, sometimes they will ask 2 more questions also. It is most common type of collenchyma which irregular arrangement and thickened angles. That is called angles that are also thickened. Where meat cells made. Example, hypodermis of tetra and nicotina. Next one, lacunar collenchyma. The collenchyma cells are irregularly arranged. That is called not regular arrangement. That is irregular arrangement. Cell wall thickened. Cell wall also thickened. The cell walls bordering intracellular space. Example, hypodermis of hypomia. Next, laminar collenchyma. Collenchyma cells are arranged compactly in layers, rows, compactly arranged. The cell wall is thickened only is tangential walls, divide of thickened or radial walls. This thickening appears of successive tangential layers. Example, hypodermis of helianthus. Next one, annular collenchyma. So that is called annular collenchyma. The report, do you know? Reported another type of annular collenchyma, petioles of nerium. The lumen is more or less circular in shape. Next one, sclerenchyma. So parenchyma, collenchyma and then sclerenchyma. Sclerenchyma is a dead cells that are both parenchyma and collenchyma is a living cells. Sclerenchyma is a dead cells and it lacks protoplasm. Protoplasm is not there. Lack means absent. The cell are long or short, narrow, thick walled and lignified secondary walls are there. The cell walls of these cells are uniformly or strongly thickened. Cell walls are uniformly and strongly thickened. Sclerenchyma cells they are 2 types. One is scleroids and another one is fibers. Scleroids is called stone cells. Sometimes ask that 1 mark questions. Which one is called stone cells? That is called scleroids. Scleroids are dead cells. Usually these are isodiametric, but some are elongated too. The cell wall is very thick due to the lignifications. Presence of the lignifications, the cell wall is thick in size. Lumen is very much reduced. The pits may be simple or branched. Pits also. The holes also is very simple or branched. Scleroids are mechanically in functions. They give hard textures to the seed coats, endosperms, etc. are present in the scleroids. Scleroids are classified into following types. Types of scleroids, very important one. There are 5 types. Brachyscleroids or stone scleroid, macroscleroid, osteoscleroids. It's called bone cells. They will ask 1 more questions. Bone cells. Osteoscleroids and trichoscleroids. First one is brachyscleroids or stone cells. Isodiametric scleroids with a hard cells wall. It It is found in bark, pit, cortex, hard endosperms, fleshy portions of some fruits also. The presence of this type of scleroids. Example, pulp of papyrus. Macroscleroids, elongated and rod-shaped cells. That is called macroscleroids. Here is the that B, elongated and rod cells scleroid. That is called brachioscleroids. Outer found in the outer seed coat of leguminous plants. Example, Crotalaria and Pisum sativums. Next one is osteoscleroid. It's called bone cells because that structure look like a bone structure. That arm bones also there, no, that structure also there. So that's why that scleroids called osteoscleroids. Rod shaped with dilated ends. The occur is leaves and seed coats. Example, seed coat of Pisums and Hackia. Fourth one is astroscleroids. That is called astroscleroids. The diagram also is very important. Star cells with the lobes are arms diverging from the central body. The occurs petioles and leaves. Example, T, nimbia and trochotendrons. Fifth one, trichoscleroids, hair-like thin-walled scleroids. That is called trichoscleroids. Numerous small angular crystals are embedded in the wall of the scleroids. Numerous small angular crystals are embedded in the wall of the scleroids. Present in the stems of leaves of hydrobites plants are present in the hydrobites plants. Example, nimbia leaf and aerial roots of Monstera. So that is called scleroids. It's called scleroid. That is called trichoscleroids. Next one is fibers, scleroids, and then fibers. Fibers are very much elongated sclerenchyma cells with pointed tips. Fibers are dead cells and have lignified walls with narrow lumens. They have simple pits. They provide mechanical strength and product from all the strong winds. The support in the provide mechanical strength of the plant. It is called supporting tissues. Fibers is called supporting tissues. Fiber have a great commercial value of cottage and textiles industries. Fibers are 5 types of fibers are there. So very important types of fibers is very important 5 mark questions. Wood fibers are ciliary fibers. These fibers are associated with the secondary xylem tissues. They are also called ciliary fibers. These fibers are associated with the secondary xylems. These are also called ciliary fibers. Secondary xylems are associated with the tissues. These tissues are called ciliary fibers. These fibers are derived from the vascular cambium. These fibers are formed in the vascular cambium. These are 2 types of there. One is limpiform fibers, another one is fiber tracheids. Second one is pars fibers or extraciliary fibers. These fibers are present in the phloems. Natural pars fibers or strong and celluloid substance are present. Fibers obtaining from the phloem or outer bark of jute, flax and hemp plants also is present in the phloems. So this is called pericyclic fiber are actually phloem fibers. Third one is surface fiber. These fibers are produced from the surface of the plant organs. Cotton and silk cottons are the examples. They are occur in the testa of seeds. They are present in the surface fibers. Next one, mesocarp fibers. Fibers obtained from the mesocarp regions of troops of like coconut. Troop coconut also they are fibers. Mesocarp fibers are present. Next one, leaf fiber. Fibers obtained from the leaf of Musa, Acave and Sansevieria. So this is called 5 types of fibers. So very important. Next one is very important that fibers in our daily life. Uses of fibers, applications of fibers, whatever they will ask. Economical uses of fibers. They will ask 5 more questions one times. I think so. Next year back, I think so. So one time they ask that question, 5 more question. Textile fibers. Fibers utilized for the manufacture of fabrics, netting and cottage etc. Next one, surface fibers. Example cottons. Soft fibers. Example flax, jute and remi. Hard fibers. Example sisal, compound, pineapple, pica, etc. Next one, brush fibers. Fibers utilized for the manufacture of brushes and brooms. Toothbrush is the that brush or washing brush, that also using this type of fibers. Rough waving fibers. Fibers utilized in the making basket, chairs, mats also etc. is produced this fiber. Rough waving fibers. Next one, paper making fibers. Wood fibers utilized for paper making. Filling fibers. Fibers used for stuffing, cushions, mattresses, pillows, furnitures. They are using that fibers is called filling fibers. Example bambax and silk cottons. That also simple tissues. Simple tissues. They are three types. One is parenchyma, collenchyma, sclerenchyma. Next one is complex tissues. The complex tissues is tissues several types of cells, but the form function together is single unit. That is called two types. So xylem and phloems. First one, xylem. Xylem means or heart room. The xylem is the principal water conducting tissue in a vascular plant. So it is a water conducting tissues. The term xylem was introduced by Negley in 1858 and is derived from xylose. Xylose means wood, Greek word, wood. The xylem which is derived from procampium is called primary xylem. So the xylem derived from the procampium is called primary xylem, and the xylem which is derived from vascular cambium is called secondary xylem. Vascular cambium is called secondary xylem. Early formed primary xylem elements are called protoxylem. So that early formed the xylem is called, the name is called protoxylem, whereas the later formed the primary xylem elements are called metaxylem. The xylem is a two types, primary xylem or metaxylems. Primary xylems means early formed primary xylems elements is called primary xylem. Lateral formed the primary xylem is called metaxylem. Protoxylem lies towards the periphery and metaxylem towards the life center is called exarch. They will ask two more questions, exarch and entarch. Entarch means the protoxylem towards the peripheral and metaxylems. So protoxylem means early formed xylems, metaxylem means later formed xylems. So protoxylem lies towards the center and the metaxylem towards the periphery. It is called, conditions called endarch. So exarch, endarch is very important. Exarch means protoxylem lies towards the peripheral and metaxylem center the center. So that is called, you see, this is called primary xylem and secondary protoxylem and this is called protoxylem, early formed protoxylem. It's a small size, that is called metaxylem. So xylem is size is increased. So protoxylem is early formed, metaxylem is later formed. The two types of xylem. Protoxylems, you see, protoxylem towards the periphery. So it is called, so some of the regions, that is called periphery regions. That is called center regions. Protoxylems lies towards the periphery. So that is called protoxylem. Towards the periphery regions is called, towards the center is called exarch. Protoxylem towards the center. So protoxylem towards the small one is in towards the center, big one is in outer side. That is called entorch. So that is called, that is a difference. Protoxylems towards the periphery and metaxylem towards the center is called exarch. So common in roots. Protoxylem lies towards the center and metaxylem towards the periphery. That's the opposite. That is called entorch. Seen in stems. Protoxylem is located at the center, surrounded by the metaxylem. So protoxylem is located in the center side. Metaxylem is called surrounded, called centrage. It is type of one vascular strand developed. Example, Selaginella species. Protoxylem is located at the center, surrounded by the metaxylems. Is called mesarch. So exarch, endarch, centrage, mesarch. This type of several vascular strands are developed. Example, obioclasm species. Next one. Xylem consists of 4 types of cells are there. One is tracheids, xylem parenchyma, vessels or trachea, and xylem fibers. There are 4 types of cells are there. First one is tracheids. Tracheids are dead, lignified and elongated cells with tapering ends are there. The lumen is broader than the fibers. Cross section the tracheids are polygonal. It's a polygonal shape. There are different types of cell wall thickening due to the deposition of secondary wall substance. Depositions of secondary wall substance, they are different types of cell are formed. One is they are annular ring-like, that is called annular ring-like structure, that is called annular type. Spiral, spring-like structure is called spiral. Next one is scleriform, it's a ladder forms are there, that is called scleriform. Next one, reticulate, net-like structure, so reticulate, and pitted, uniformly thick except at pits. Tracheids are imperfected cell wall broadened pits on the side wall. Only through these conditions they takes place in a gymnosperms, only the pitted tracheids are formed. They are arranged one above the others are arranged other. Tracheids is a chief water conducting elements in gymnosperm and teratophytes. They are also of mechanical support of the plant. So tracheids, gymnosperms and teratophytes, they are both main chief water conducting elements. They are also offer mechanical support to the plant. Next one, vessels. So it's called trachea. Vessels are elongated tube-like structure. They are dead cells formed from row of vessels element placed on one end to end. They are placed. They are perforated at the end wall. Their lumen is wider than tracheids. Due to the dissolutions of entire cell's wall, a single pore is formed at the perforations plate. It is called the simple perforations plate or there. Example, Mangifera. The perforation plate has many pores. So single, only one pores are there means simple perforation plate. Their perforation plate has many pores are there. It's called multiple perforation plate. Example, Liriodendron. The secondary wall thickening of vessels are annular, spiral, scleriform, reticulate or pitted as a tracheids. Vessels are chief water conducting element in the angiosperms and absence in teratophytes and gymnosperms. The vessels is not there. Only the presence in only for angiosperms, not for teratophytes and gymnosperms. The teratophyte gymnosperms are absent in vessels. Netums of gymnosperms vessels are occur. The main functions of conductions of water, minerals and also offers mechanical strength. Next one, xylem fibers. Xylem fibers are sclerenchyma associated with the xylem are known as xylem fibers. Xylem fibers are dead cells and have lignified walls with narrow lumens are there. They cannot conduct water, but being stronger provide mechanical strength. They will not conduct the water, but stronger provide mechanical strength. They are present in the both primary and secondary xylem. Xylem fibers are also called libriform fibers. It's called libriform fibers. The fibers are abundantly found in many plants. They occur in patches, in continuous bands, and something singly among the other cells are there. Between fibers and normal tracheids, there are many traditional forms which are neither typical fibers or typical tracheids are there. The transitional types of design as fiber tracheids. The pits of fiber tracheids are smaller than those of vessels and typical tracheids. They are fibers. Next one, xylem parenchyma. Xylem parenchyma, the xylem parenchyma cells associated with the xylem are known as a xylem parenchyma. So xylem parenchyma cells associated with the xylem is called xylem parenchyma. These are the only living cells in the xylem tissues. So xylem tissue, all the tissues are dead tissue only for living tissues, the parenchyma tissue. The cells wall thin and made up of cellulose. Parenchyma arranged longitudinally along the long axis called axial parenchyma. Ray parenchyma is arranged in the radial rows. Secondary xylem consists of both axial and ray parenchyma. Parenchyma stores food materials and also help in the conduction of water. That is the parts of xylem parenchyma. So xylem. Next one, phloem. Phloem to leptome. Phloem is the food conducting complex tissues of vascular plants. The term phloem was coined by Negley 1858. The phloem which derived from procampium is called primary phloem, and the phloem which is derived from vascular chemy is called secondary phloems. Early formed primary phloem elements are called protophlloem. So same like that, xylem also that same also. Whereas the lateral formed the primary phloem elements is called metaphloems. Protophlloem is short-lived. It gets crushed by the developing metaphloems. Phloem consists of 4 types of cells: sieve elements, companion cells, phloem parenchyma and phloem fibers. First one, sieve elements. Sieve elements, they are conducting elements of the phloem. There are 2 types only, name is sieve cells and sieve tubes are there. Sieve cells, these are primitive type of conducting elements found in pteridophytes and gymnosperms. They are present in pteridophytes and gymnosperms. Sieve cells have sieve areas on the lateral walls only. They are not associated with the companion cells. Sieve tubes. Sieve tubes are long tube-like conducting elements in the phloem. These are formed from the series of cells called sieve tube elements. The sieve tube elements are arranged one above the other form of vertical size tube. So one above the one is are arranged. So that vertical size tube, sieve tube. The end of wall conducting number of pores and look like a sieves. So it is called sieve plate. So number of pores are there, that is called sieve plate. The sieve element shows necrosis thickening on the lateral walls. They may be possess simple or compound sieve plates are there. The functions of sieve tubes believed into the controlling the companion cell, that is a functions. In mature sieve tubes, nucleus is absent. It contains lining layer of cytoplasms are there. Special proteins called slime body is seen in the sieve elements. In the mature sieve tubes, the force is the sieve plate are blocked by the substance called callose. They will ask 2 more questions. Mature sieve tubes is that sieve tubes pores, many pores are there. So sieve plate. So many pores are there. That is called sieve plate. The sieve plate blocked the substance. So it is called callose. The conductions that is called sieve plate. So many sieve plates are there. The substance enter to the substance, that is the substance will not go because that sieve plates are blocked that area, that is called callose. Through cytoplasmic strands, sieve tubes occurs only the angiosperms. Next one, you see that is called sieve tubes, sieve plate. Center, that is called campion cells. Campion cells, the thin-walled, elongated, specialized parenchyma cells which are associated with the sieve elements are called companion cells. So parenchyma cells associated with the sieve element, that is called companion cells. These cells are living and they have cytoplasm and prominent nucleus. They are living under cytoplasm, so prominent nucleus are present in the cells. They are connected to the sieve tubes through the pits found in the lateral walls. Though these pits, cytoplasmic connections are maintained between these elements. The cells are helpful in maintaining the pressure gradient in the sieve tubes. Usually the nuclei of the companion cells serves as the nuclei of sieve tubes as they lack them. The companion cells are present only in angiosperms. and absent in gymnosperms. The companion cells only for angiosperms only, not for gymnosperms and tetraphytes. They assist the sieve tubes, the conductions of wood material. Next one is phloem parenchyma. The parenchyma cells associated with the phloem is called phloem parenchyma. So parenchyma associated with the phloem, that is called phloem parenchyma. These are living cells. They are store starch and fats. They also contain resins and tannins in some plants. Resins and tannins are some plants are stored in the starch and fats also. Primary phloem consists of axial parenchyma and secondary phloem consists of both axial and ray parenchyma. Both are there. They are present in the teratophyte, gymnosperms and dicot. Next one is phloem fibers are bast fibers. Fibers of sclerenchyma associated with the phloem. So sclerenchyma associated with the phloem is called bast fibers or phloem fibers. They are narrow, vertically elongated cells which are thick-walled and small humans. Among the 4 phloem elements are there. Phloem fibers are only dead cells. Phloem only for dead cells. Phloem fibers only dead cells. Remaining all the phloem is lived elements, tissues. They are the strengthening as well as supporting cells. That is called plant tissues. Overall concept map. So they are plant tissues. They are 2 types. One is meristematic tissues, another one is permanent tissues. Meristematic tissues capable active the cell divisions. They are based on the positions, origin, based on the function, based on the division. They are 4 types of the permanent tissues. Lost the power of cell divisions. So cell division is not occurring here. So they are 2 types. One is simple tissues and another one is complex tissues. Simple type tissues means only one type of tissues. Complex tissues means more than one type of cells. That is called complex tissues. The simple tissues, they are 4 types. Parenchyma, collenchyma, sclerenchyma. 3 types are there. Parenchyma, collenchyma, sclerenchyma. Parenchyma, they are 5 types of the parenchyma, storage parenchyma, stellate parenchyma, chlorenchyma, prosenchyma. Collenchyma means they are 3 types: angular collenchyma, lacunar collenchyma, laminar collenchyma. Sclerenchyma, dead cells and lignified wall. They are 2 types. One is scleroids and fibers. Scleroids, they are 5 types: branchyscleroids, macroscleroids, astroscleroids, trichoscleroids. They are very important. That scleroids. Fibers, they are fibers: food fibers, past fibers, surface fibers, mesofibers, leaf fiber. They are 5 types. That examples also is very important. Next one, complex tissues. So more than one type of cell. There are 2 types: xylem and phloem. Xylem, so water conducting tissue. Phloem is a food conducting tissues. Tracheids, they are 4 types: tracheids, vessels, xylem fibers, xylem parenchyma. So tracheids is a dead cells, vessels is a dead cells, xylem fibers also dead cells, lignified and sclerenchymatous. Xylem parenchyma is a living cell. That living cells only for xylem parenchyma. They will ask one more question. It's very important. That same phloem also conducting, it's a food conducting tissue. They are 4 types of the sieve elements. Is a sieve tubes, companion cells. Phloem parenchyma is a living, is a absent in monocot. Phloem fibers, thick-walled, sclerenchymatous, giving mechanical strength. They are phloem parenchyma is a living cells. So that is a flow chart of that complex permanent tissues and meristematic tissues. Next one, different type of tissues: parenchyma, collenchyma, sclerenchyma. They are same like that. The distributions, main functions, nature, cell type, wall materials. For example, parenchyma, cortex, pith, medullary rays and packing tissues or vascular systems also the main functions. Packing tissues support gases exchange, food storage. Living tissues, cell type usually isodiametric. Wall materials mainly cellulose and pectins are there. Collenchyma, so out regions of cortex, angles of stems, midrib of leaves, they are present in the collenchyma. So it's a main functions, mechanical support. It's a living cells, elongated polygonal, mainly cellulose, pectin and hemicellulose are present in the cell wall. Next one, Sclerenchyma, they are two types. One is fiber, another one is scleroids. Fibers, the out regions of cortex, pericycle stems, vascular bundles are there. Mechanical support, so give to functions, mechanical support, dead cells. It's called dead cells. Elongated and polygonal, tapering the mainly lignins, the cell walls. The materials is called lignins, presence of lignins. Scleroids, cortex, pith, phloem cells and stones of fruits and seed course, mechanical protections. Dead cells, roughly isometric with much variations, mainly lignins. Next one, tracheids and vessels, vascular system, translocation of water and mineral salt, that is a functions. That also dead cells, elongated and tubular, mainly lignins are present. Next one, phloem sieve tubes and accompanying phloem sieve tubes, vascular systems, translocation of organic solids, living tissues, elongated and tubular, cellulose, presence of the cell wall, cellulose, pectin, hemicellulose are present. Companion cells, vascular systems, work in association with the sieve tubes, living, elongated and narrow, cellulose, pectin and hemicellulose are present. So next one, that difference also is very important. Meristematic tissues, permanent tissues. Cell divided repeatedly, you do not divide. Cells are undifferentiated, cells are fully differentiated. Cells are small or isodiametric, the variable in shape and size. Intracellular space are absent, intracellular space are present. Vacuoles are absent, vacuoles is not there, so vacuoles are present. Cell wall are thin, cell walls may be thick or thin. Inorganic inclusions are absent, inorganic inclusions are present. That is difference between meristematic tissues and permanent tissues. Next one, collenchyma and sclerenchyma. They will ask 2 mark, 3 mark or 5 mark questions. All the differentiations is very important. Living cells, say dead cells containing protoplasm, do not have protoplasms. Cell walls are celluloid, cell walls are lignified. Thickening cell walls is not uniform. So cell wall is uniform. Keeps of the plant body soft. The keeps plant body stiff and hard. The scleroids present in this sclerenchyma, the plant body is very hard. Sometimes it has chloroplast. Do not have chloroplast. That is important. Next one, fibers. Difference between fibers and scleroids. Long cells, so short cells, narrow, elongated, pointed ends. Usually short and broadened. Occurs in bundles. Occurs individually or in small groups. Commonly unbranched, so may be branched. Derived directly from meristematic tissues. They will develop from secondary scleroids or parenchyma cells. Next one, difference between tracheids and fibers. Not much tracheids, not much elongated, very long cells. Possess oblique and end walls, very long cells. Possess tapering end walls. Cell walls are not as thick as fibers. Cell walls are thick and lignified. Possess various type of thickening. Possess only pitted thickening. Responsible for the conductions and also mechanical support. Provide only for mechanical support. That is difference between tracheids and fibers. Next one, sieve tubes and sieve cells and sieve tubes. Have no companion cells, have companion presence of the companion cells. The sieve areas do not form sieve plates. They do not form sieve plates, but here they confine the sieve plates. The sieves areas not well differentiated. The sieve areas are well differentiated. They are elongated cells and are quite long with tapering end walls. They consist of vertical cells placed one above the other, forming the long tubules connected walls or sieve pores. The sieve are smaller or numerous. The sieve pores are longer and fewer. Found in teratophytes and gymnosperms. Found in angiosperms. That is difference between sieve cells and sieve tubes. So now class is over. Now I will give you that today test. Explain the shoot apical meristem theory. That all the theory, apical cell theory, histogen theory, tunical corpus theory, that is also is very important. You have to write separately. They explain the root apical meristem theory, apical cell theory, and diagram. That diagram also is very important. Histogen theory and corpus cap theory. Next one, give account on parenchyma, collenchyma and sclerenchyma. The types also is very important. So all the three is types, that 5 types, 3 types, everything is very important. Next one, explain the types of fibers, types of fibers, that 5 types of fibers is very important. Next one, explain the fibers in our daily life. So what are the uses of economic importance of fibers, that is very important. Give account on xylem tissues, that 4 tissues are very important. Next one, give account on phloem tissues, it's a very important. So today test, so that all the questions is very important, that lesson, that chapter also is very important. Definitely they will ask 5 more questions. Okay, today class over, so we will see the next class. Thank you.